Hello, this is Julieta Rey, and in this video I will be explaining membrane-bound receptors, soluble receptors, gated ion channels, RTKs, second messengers, and G-protein coupled receptors. This picture right here signifies the membrane-bound receptors. This is just an idea, and later on in the video I will go further into detail. So these receptors are hit by ligands, which is a signal to open a gated channel, and lets ions go through the two bilayer membrane and into the cytoplasm. Detailed picture of a membrane-bound receptors and how they work. So the G-protein coupled receptors are bound to the membranes. When a ligand makes contact with it, it inactivates this G-protein, which makes the alpha subunit float away and later travel to the effector protein to activate cellular response for genes or into the endoplasmic reticulum. So after this alpha subunit floats away, GDP is converted to GTP, and GTP happens again, makes the alpha subunit float away and attaches to the effector protein, GTP, and the alpha unit. Although this picture is very messy with my notes, I will still be able to fully introduce and explain CAMP-mediated signal transductions. So as in the clip before, the G-protein coupled receptor makes contact with the ligand, and GDP attaches to the alpha, beta, and gamma subunits, which later makes the alpha subunit float away, attached to the adenylyl cyclase, which is an enzyme, and it turns it on, uses ATP to make CAMP, which is a second messenger. CAMP changes and activates pKa, which catalyzes it, and it reaches a response protein, which then later makes a cellular response. CAMP will also turn on pKa, which is a kinase, which makes phosphates to proteins by phosphorylation. This picture depicts a calcium IP3 gated mediated signal transduction. As shown in the previous videos, the ligand makes contacts with the G-protein coupled receptor, which activates GDP to GTP to make the alpha subunit float away, attach itself to phospholipase, and make PIP2 to DAG and convert it to the second messenger IP3, which is then used with calcium to make a cellular response and open an IP3 calcium channel, which forces out calcium into the cytoplasm. It is also a kinase, which is almost of the same idea. It makes contacts with the membrane receptors, activates, and phosphorylates each other, as shown in this image. I hope this video was understandable. Thank you.